Hello everyone, I'm Sai Shraddha and today I'll be talking about the genomic dataset curation and evaluation of the DNA-BIRD S model, which is a foundation model for resistance prediction task in tuberculosis. The reason we chose to go for tuberculosis is that it's one of the deadliest bacterial diseases out there and it causes a lot of highest rates of death. And most importantly, over the time, the resistance to drugs is increasing. So we don't know which drugs are going to work against newly incoming strains or not. And clinically diagnosing this thing, whether an incoming strain is resistant or not, takes very, very long time. So it's very natural to dive into machine learning techniques to do this resistance prediction tasks because it's extremely faster that way. But it's not very straightforward because this machine learning techniques have not been applied so much in the bacterial genomics like they have been in human genomics. So definitely the data sets are extremely scattered. It's of a very, very bad quality and it's not directly available in the format that will be useful to apply machine learning. And moreover, the studies that have tried applying machine learning for TB, especially for resistance prediction, build their own custom pipelines, their own custom data set, and then they perform modeling on that. So that way, all these pipelines and datasets cause heterogeneity across different methods. And this is why the performances of one model or one method cannot be directly compared with another method. It's not an apple to apple comparison. This is why we aimed to try to build a standardized unified pipeline and a dataset, somewhat inspired by ImageNet, to make this evaluations across different models very fair. And so once we have this dataset, we also wanted to see how good the existing machine learning techniques that have been tried to be applied to TB or have been kind of trained on TB are good at predicting resistance or sensitivity to a given drug or not, given an incoming strain. The third one was that we also tried to build an interpretability dataset, which essentially acts as a ground truth table or a ground truth data that lists down not just whether an incoming isolate can be resistant or sensitive to a drug, but also like which particular mutations in that strain can cause resistance to a drug, can be responsible for resistance to a drug. So this is important because we also want our models to identify the most important mutations that actually cause resistance and not just be good at predicting whether they are good at resistance or not. And this is extremely important in biology. Just an idea of how the interpretability dataset looks like here, given a strain, there are different mutations that are taken within that strain, and then we label each of these mutations with our resistance or sensitive to a drug or not. An idea of the data unification pipeline. So our models, machine learning models, need DNA sequences for their input. So we take variant calling files, which are essentially for a given strain. It lists down what mutations have taken place with respect to the reference strain in this new isolate. So using these mutations, we reconstruct our new isolate or new strain. So we reconstruct the DNA sequence for this mutated strain, and this acts as the input for our benchmarking machine learning models. The second one is the interpretability dataset. It goes through a lot of pre-processing that I have to do, and then finally I create the label of mutation with respect to the phenotype for a given drug, and this is the basis of the interpretability and serves as the interpretability dataset for future directions. So for benchmarking, our task is to predict whether for a given variant or a given DNA strand, it is going to be resistant or sensitive to a drug or not. So we try 3 different models from different genres. One is the simple machine learning technique. We use logistic regression for it. So we take a DNA strand for a given variant, pass it through the model, and for a given drug we predict the resistance or sensitive label. And three different, so for every different drug, every different logistic model is trained. So if we have 11 drugs, 11 different models will be trained. So it's a single task model. Then we go to a little complex model, which is a CNN model, multi-drug CNN model, which essentially, instead of just being trained on a different drug one at a time, it's being trained at for all the drugs one at a time. So it's a multi-task model. This model doesn't take in the whole DNA strand, but we pick out important regions on the DNA that are known to be causing resistance to all these drugs, and then one hot encode these regions and then stack these regions over each other and then pass to the CNN model. So the one hot encoding is done in a way that, like if you see this matrix on the left side, there can be 5 possibilities at a given position on DNA: A, T, G, C, and then a dash. So if A occurs at that particular position, it's marked as 1, and rest of the letters are marked as 0. And the same thing is done for each of the positions on the gene. This kind of gives a 2-dimensional matrix for one gene region and all the different gene, and this is done for 11 different gene regions. So they are stacked over each other and passed to the CNN to do predictions of resistance or sensitivity. This is the architecture of the CNN module. The third one is the foundational model. We use DNA-BERT-S model, which is known to have been trained on bacterial genomic data. So we do think that it has some idea of how the genomic data looks like and what the underlying mechanisms should be and stuff. So this is why we choose to go for this model. So here again we choose important genomic regions from the DNA strand, and each of these genomic regions are tokenized using byte pair encoding and then passed to the DNA-BERT-S, and then we generate embeddings for each of these gene regions. Again, like we did in CNN, we stack each of these embeddings one over each other and then pass through a downstream small CNN model to do the resistance prediction. Again, this model is a multi-task model like the MD-CNN previously. We use pre-trained DNA-BERT-S, and again we also fine-tune DNA-BERT-S for resistance prediction to see whether the performance changes or not. This is an overall architecture of DNA-BERT-S. Looking into the results, starting with AUC, well, the red one shows DNA-BERT-S, the blue one is MD-CNN, and the gray one is logistic regression. It's very surprising to see that DNA-BERT-S fails. And this kind of behavior is shown for both pre-trained as well as fine-tuned models, which is extremely weird because they are known to perform well in general tasks. And given that DNA-BERT-S was pre-trained on tuberculosis data also, it should have been able to do at least better than what it does right now. So we see a very random AUC. That means that the model is not actually learning anything. It's just performing randomly. It's not able to differentiate between resistant or sensitive isolates. If we look into sensitivity, the same thing is shown because the behavior of DNA-BERT-S model is extremely random. That means that it is not able to predict well whether a given strain is resistant or not. This kind of tells that the embeddings are not of that good quality, because I also tried to apply multiple different architectures of CNN with different settings of hyperparameters, starting from shallow to deeper, even with MLP, but none of the models were able to generalize well on these embeddings. That means that the embeddings themselves are not enriched enough for resistance prediction. This points to the idea of negative transfer, that tells that a model that has been pre-trained on some kind of other task before, and it's trying, and the generated embeddings are being trained to perform some other kind of task, it will hurt the performance of the downstream models. And this is what we see here, because DNABERT-S was primarily trained for clustering of similar species. So for resistance prediction task, it terribly fails because it's not able to identify or learn important mechanisms for predicting resistance like DNABERT-S, like CNN or the logistic regression does. So that means that first of all, it's very important for embeddings to be enriched that come out of foundational models. So in order to do predictions, good predictions in biology for a given task, the models, it's not necessary that the models just have to be pre-trained on a lot of big data on similar kind of species, but they have to be pre-trained or at least trained on similar kind of task, or they have to be injected with more biological information for them to perform really good on specific biological tasks, which is unlike what we see in other general CS domains. And this has been an interesting study, and I will study the interpretability part of this also going forward. Yeah, so this was an overview of my project. Thank you.